This video is my last video of IP8 and will cover some skeletal muscle relaxants. Some of the drugs in this section will be a review of previous drug classes. Some will be new, and I also realize this is quite random where it falls, but the issue with some of these skeletal muscle relaxants is that they don't neatly fall into any one pharmacologic class or disease state. I will spend most of the time talking about neuromuscular junction blockers or NMJ blockers, and then we'll mention briefly a few other drugs that relax skeletal muscle or are what we consider spasmolytics or antispasmodics because they reduce muscle spasms. Here are the muscle relaxants I will cover. As I mentioned, I will spend most time talking about neuromuscular junction blockers, but we'll also briefly touch on dantrolene, baclofen, diazepam, tizanidine, cyclobenzaprine, and botulinum toxin, or Botox. The neuromuscular junction blockers are, as their name suggests, drugs that block synaptic transmission or activity at the junction between neurons and muscle. The physiologic effect of this activity is to produce total muscle paralysis throughout the body, but without significant effects on CNS or cognitive function or on the cardiovascular system, because these drugs have little to no activity in the central nervous system or at autonomic ganglia, which controls cardiac and other organ function. The main reason, therefore, that these agents would be used is during surgical procedures and in the ICU to produce intentionally some muscle paralysis. This can be effective when used as an adjunct during general anesthesia to relax skeletal muscle. This is especially useful during abdominal surgeries so that there's not contraction and spasms of skeletal muscle in the abdomen during abdominal surgery, as this can cause problematic injuries during a surgical time. In addition, these agents can be used to facilitate endotracheal intubation because they relax the skeletal muscles of the trachea and the lung. The potential problematic effect of total muscle paralysis, however, is that there can be respiratory paralysis, where the respiratory muscles surrounding the lungs become fully paralyzed and the individual becomes incapable of breathing. Therefore, these agents typically need to be used with some sort of artificial respiration or used at a concentration that does not produce respiratory paralysis. Those aspects of these drugs would be discussed whenever they come across in a clinical situation during a therapeutics or active learning session. As I mentioned, these agents have virtually no CNS activity because they do not cross the blood-brain barrier. They have only very minor cardiovascular effects. The most likely one that would be seen is some increased heart rate. In addition, many of these drugs, because of their structure, are able to induce histamine release from mast cells, which is due to a direct action on the mast cells. And this can cause some itching, rash, and some allergic type reactions. The mechanism of action of these drugs is that they block nicotinic cholinergic receptors at the neuromuscular junction. These are the NM type nicotinic receptor because they are found in muscle, and they have a specific receptor subtype conformation of two alpha 1 subunits, a beta 1 subunit, an epsilon, and a delta subunit, as described in the text in the table, but also shown graphically below in the muscle conformation. Nicotinic cholinergic receptors are pentameric ion channels that allow sodium to flow through the channel when they are open. The endogenous agonist or stimulating agent for these receptors is acetylcholine, whereby it opens the channel, allows sodium to enter, and therefore conduct an action potential or cause a muscle contraction. The neuromuscular junction blockers specifically inhibit the NM subtype of the nicotinic receptor, which is found exclusively at the neuromuscular junction. These are drugs like atracurium, vecuronium, and pancuronium. There are other two primary subgroups of nicotinic receptors. These are the neuronal nicotinic receptors, or NN type, found in autonomic ganglia, and the central neuronal type, or CNS type, found in the brain. Acetylcholine does activate these different subtypes of nicotinic receptor, but the neuromuscular junction blockers we are going to talk about now have no effect at either the autonomic ganglia or CNS subgroup of nicotinic receptor. The neuromuscular junction blockers can be subdivided into two different classes based on their pharmacologic activity. These include the depolarizing blockers, which there is one type I will mention called succinylcholine, or the competitive or non-depolarizing subgroup, which includes pancuronium and most other neuromuscular junction blockers. I will discuss their mechanisms of action separately. First, I will mention the depolarizing blockers, like succinylcholine. These are drugs that are competitive antagonists of acetylcholine binding to the NM receptor. As demonstrated in this image, the depolarizing blocker in blue competes with acetylcholine for binding to the channel. It initially opens the channel, but keeps it open for an extended period of time, which results in eventual blockade of sodium flow. This long-term opening of the channel is termed depolarization and results in the channel becoming inactive long-term, which produces the paralytic effect that we see with these drugs. Therefore, the physiologic effects of depolarizing agents are to produce an initial muscle excitation followed by prolonged paralysis. As demonstrated in this image, the depolarizing blocker succinylcholine keeps the channel open but blocked and therefore inactive. In contrast, the non-depolarizing agents do not block the channel, but instead keep the channel in a closed conformation. Again, these drugs are competitive antagonists of acetylcholine binding to the receptor. They compete with acetylcholine for binding to the NM ion channel, and they keep the channel closed and prevent acetylcholine from binding at the same time. This means that the channel remains in a closed conformation and does not allow ions to flow through, thereby leading to extended muscle paralysis. One main difference between the depolarizing and the non-depolarizing drugs is that the non-depolarizing agents can be reversed by sufficient concentrations of acetylcholine, and therefore their effects can be reversed or treated by the use of acetylcholinesterase inhibitor. Another main difference between the depolarizing and the non-depolarizing drugs is their structure. Succinylcholine is the only depolarizing blocker, and it bears significant structural resemblance to the endogenous molecule acetylcholine. It is essentially a double acetylcholine structure, where two acetylcholine molecules are linked together end to end. It's a relatively flexible structure that connects two quaternary ammonium heads with two positive charges on the nitrogen ions. This results in a molecule that is very poorly lipid soluble and is not able to cross the blood-brain barrier. The non-depolarizing agents also include a double acetylcholine structure, but in this case, it is concealed within some larger ring system, either an isoquinoline ring system or a steroid ring system. As demonstrated on your screen, the drug atracurium is an isoquinoline type structure that contains two molecules of acetylcholine joined end to end with the quaternary ammonium ions and larger ring substituents. The steroid-based ring structures include all the other non-depolarizing agents, such as pancuronium and vecuronium. The main center of the molecule is a steroid ring structure with two acetylcholine molecules concealed in the drug structure, as evidenced by the blue line in pancuronium. The non-depolarizing drugs are much more rigid than succinylcholine, and therefore they tend to have fewer off-target effects at other receptors. These molecules are also very poorly lipid soluble due to their two positive charges and are not able to cross the blood-brain barrier. In terms of the kinetics of these drugs, as I mentioned, given their positive charges, they are highly polar and for that reason are inactive orally and do not cross the blood-brain barrier. However, given their solubility in water, they are well distributed by the IV route. In terms of their metabolism and elimination, their duration is limited by hepatic hydrolysis by various enzymes. These drugs can be classified into one of three groups based on their duration of action. Succinylcholine is considered an ultra short duration at only 6 to 11 minutes. It is eliminated by plasma cholinesterases and therefore does not require hepatic or renal elimination mechanisms. Rocuronium, vecuronium, and cistercurium are all considered intermediate acting based on their duration of 30 to 90 minutes. Rocuronium is eliminated by hepatic hydrolysis and therefore is problematic in liver dysfunction. Vecuronium is eliminated by both hepatic hydrolysis and renal elimination, and therefore is problematic in either liver or kidney dysfunction. Cistercurium has an interesting elimination or metabolism scheme, which includes a Hoffman elimination reaction. This makes it possible to administer cistercurium to someone with liver or renal dysfunction because it does not require hepatic or renal elimination. Cistercurium is also a steroid-based ring structure. Finally, pancuronium is considered a long duration blocker at 90 to 100 minutes in duration of action. It is eliminated by renal and hepatic mechanisms and therefore may not be used if significant kidney or liver dysfunction. One additional note about the activity of succinylcholine and pancuronium is that both of these drugs do have some stimulation of muscarinic receptors, which result in some muscarinic side effects like tachycardia or increased intraocular pressure in the eye. However, given their relatively short duration of action and their use primarily as short-term anesthetic adjuvants, this may not be an issue for most individuals. Now I will briefly mention some other skeletal muscle relaxants that act at various areas both in the central nervous system and at the neuromuscular junction or within the muscle itself. The first drug I will mention is dantrolene. Dantrolene acts locally within the muscle itself to block calcium release channels in the sarcoplasmic reticulum of skeletal muscle. If calcium cannot be released from the sarcoplasmic reticulum of skeletal muscle, the muscle cannot contract and it becomes relaxed and spasms are reduced. The next drug I will mention is baclofen. Baclofen is an agonist at the 7-transmembrane GPCR GABA B receptor, which is a second type of GABA receptor that is considered a G protein coupled receptor. Stimulation of the GABA B receptors in the spinal cord facilitates spinal inhibition of motor neurons. This results in the motor neurons becoming essentially inactive and not able to stimulate skeletal muscle, therefore also reducing muscle spasms and relaxing muscle. Diazepam has a similar effect in the spinal cord. Diazepam facilitates GABA transmission in the CNS and the spinal cord to again inhibit spinal motor neurons. The next drug is tizanidine. Tizanidine is an alpha-2 adrenergic receptor agonist, also in the spinal cord, and this again has activity to inhibit spinal motor neurons. Next, I will mention botulinum toxin. Botulinum toxin inhibits the release of acetylcholine through an activity on the process of exocytosis of vesicles containing acetylcholine at the neuromuscular junction. By inhibiting acetylcholine release, This prevents acetylcholine from acting on skeletal muscle and therefore causes muscle paralysis at the neuromuscular junction. However, Botox can also cause other effects in different parts of the body where acetylcholine is released, such as in autonomic ganglia, in the CNS, and at parasympathetic effector organs. Infection with botulinum toxin can cause certain death because of these widespread and significant effects. However, as you know, Botox is used as a muscle relaxant and a muscle paralytic when injected into the face, for example. And the main mechanism by which it does that is inhibiting acetylcholine release onto the skeletal muscle of the face. This is maintained as a local action because the botulinum toxin or Botox that is injected into the face is inactive and not able to affect other parts of the body. Finally, the last agent I will mention is cyclobenzaprine. Cyclobenzaprine is a drug with anticholinergic, antimuscarinic, and antihistaminic activity. It has a very poorly understood mechanism of action to relax skeletal muscle, but is thought to centrally inhibit the muscle stretch reflex by acting at the level of the spinal cord. It is not used as frequently as a skeletal muscle relaxant, given the availability of other agents with fewer side effects. The image on your screen also illustrates the effect of botulinum toxin at the neuromuscular junction. You can see at the NMJ where acetylcholine is released to act on skeletal muscle. It is at this interface that botulinum toxin inhibits acetylcholine release and prevents muscle contraction from occurring. At this point, it would be a good idea to review the summary table at the very end of Chapter 27 in the Basic and Clinical Pharmacology textbook. If you take a photo of the QR code on the screen, that will bring you right to Chapter 27 of the text. The summary table in Chapter 27 describes the activity of all of the skeletal muscle relaxants I have just mentioned and provides a very good overview of the spasmolytic and antispasmodic drugs.